Good day, I am Andriana Merced, a first-year nursing student here at UCQ, belonging to the LO2 class. Today, I will be discussing a poster that I have made which will be answering the question, how does the role of gut microbe contribute to human health? We will first go over a brief introduction to the topic, then discuss the functions of the gut microbe. Afterwards, we will discuss what microbial dysbiosis is and how it affects us. And lastly, the conclusion. Our guts are considered a separate entity within the human host due to the colonization of a complex microbial system. It is definitely a busy and crowded metropolis of interacting microbes. Over the past few years, we have seen a multitude of extensive research done to establish the involvement of human gut microbiome and several aspects of human health. Van de Wiel et al. 2016 reported that the intestinal tract is a nutrient-rich environment that contains up to 100 trillion microorganisms, most of which reside in the colon. In the human gut, over 50 bacteriophyla have been detected, but normal microbiota is often dominated by anaerobic bacteria and two phyla, the gram-negative Bacteroidetes and the gram-positive Firmicutes. Bullen-Plummer 2014. Other identified species in the gut microbiota have specific functions in the gut ecosystem. Now we will move on to discussing the functions of the gut microbe. First one is the role in gut-brain axis. According to Bullen-Plummer 2014, this is defined as the bidirectional communication system that encompasses neural, hormonal, and immunological signaling between gut and brain. Zaidan and Cryan 2017 stated that the intestinal microbiota communicates with the brain using different routes, including the vagus nerve, short-chain fatty acids, cytokines, and tryptophan. Through these routes, gut bacteria can produce neuroactive chemicals and regulate key central neurotransmitters by altering levels of precursors. Furthermore, Van de Wiel et al. 2016 reported that the pattern recognition receptors identifies if the immune system is activated or regulated by distinguishing trustworthy microorganisms and opportunistic pathogens. Number 2. Protection from colonization by pathogenic species. According to Bullen-Plummer 2014, the barrier or competitive exclusion effect occurs when the intestinal bacteria produce antimicrobial compounds and fill in available functional places. This allows intestinal bacteria to compete with pathogens for nutrients and sites of the attachment in the gut lining. Additionally, other intestinal bacteria are capable of killing or inhibiting other potentially harmful pathogens, or denature bacterial toxins by producing substances such as non-specific fatty acids to highly specific bacteriocins. Bullen-Plummer 2014, Quigley 2013. Third is supporting and priming the immune system. The development of a host immune system is dependent on the dynamic and continuous interactions of the intestinal microbiota and its metabolites. It influences the maturation and development of lymphoid tissues. Quigley 2013 stated that the Bacteroides fragilis can correct T cell deficiencies and Th1 and Th2 imbalances, and direct the development of lymphoid organs in the germ-free organism. It can also regulate production of immune mediators such as immunoglobulin A. Dendritic cells can modulate T cell activation and maturation into specialized functional subsets upon antigen recognition. This will help activate B cells and produce immunoglobulin A, which has a major role in antimicrobial protection within the intestine. Gut microbes can also introduce low-grade inflammatory response. Quigley 2013 reported that the introduction of low-grade inflammatory response allows our body to deal with other pathogens more aggressively in the future, therefore priming our immune system. Certain bacteria can differentially induce regulatory T cells and result in the production of the anti-inflammatory cytokine interleukin. Number 4 is the role in metabolism. According to Bullen-Plummer 2014, gut bacteria are capable of producing enzymes to digest and harvest energy from indigestible carbohydrates, unabsorbed dietary sugars and alcohols, as well as some oligosaccharides that escape digestion. Other bacteria can also produce several vitamins and minerals needed to survive, such as folate, thiamine, and vitamin K. Additionally, gut bacteria can also synthesize all essential and non-essential amino acids. Now we will move on to discussing what is microbial dysbiosis and how does it affect us. Having a drastic change in the gut microbiota or a lack of diversity can lead to several diseases, syndromes, and functional abnormalities. First, we will be discussing the systemic metabolic diseases. Diabetes. Specific microbial components contribute to the regulation of energy metabolism as well as in glucose and lipid homeostasis. Kani 2018 stated that the constituents of gram-negative bacteria such as lipopolysaccharides were the key factors in triggering the onset of low-grade inflammation and insulin resistance via mechanisms of interaction between gut microbe and the innate immune system. Second is obesity. Obese adults tend to have higher levels of short-chain fatty acids compared to lean individuals due to the change in fecal flora. Bull and Plummer 2014. Additionally, Quigley 2013 reported that further studies have indicated that the microbiota shown on obese individuals stimulate hepatic triglyceride production by suppressing a lipoprotein lipase inhibitor, which leads to a continued expression of LPL, a key regulator of fatty acid release from triglycerides in the liver. Now we'll be discussing the chronic gastrointestinal diseases. Irritable bowel syndrome falls in this category. According to Quigley 2013, IBS patients tend to have a leaky epithelial barrier due to a decreased fecal flora, which allows bacteria to enter the submucosal compartment, where activated lymphocytes and mast cells reside. This occurrence will activate sensory neurons and can result in local reflexes that affect motor and secretory functions or even lead to enhanced visceral sensations centrally. Inflammatory bowel disease also falls under this category. This occurs when there is a reduction in diversity and abundance of the Firmicutes population. Firmicutes is a bacteria that produces short-chain fatty acids and are known to have anti-inflammatory properties. Bull and Plummer 2014. C would be allergic diseases. Bull and Plummer 2014 reported that the low exposure to Proteobacteria during infancy will lead to a higher risk of atopic eczema. In this study, it was uncovered that underexposure to microbial antigens result in the aberrant outcome to allergen processing of immunological response rather than immunological tolerance. Now finally, we will move on to the conclusion. Diversity is the key to a healthy microbiome. Several studies show countless positive functions and play key roles in metabolism, immune response, protection from pathogens, and in gut-brain communication if it's in a healthy state. We need our gut microbes just as much as they need us to keep us healthy and resilient to drastic changes. As more studies continue to be published, our knowledge regarding this complex world continues to broaden. Through this, we are able to understand the vital role our gut microbes have in shaping our health, well-being, and even in our personal identity. Thank you for listening.